All right, welcome to part 2 of the Human Microbe Interaction lecture on Chapter 13 material. So the source of microorganisms is anywhere in the environment. They can be present inside of a living host or in a non-living element of the environment. And these areas where microbes are harbored are called reservoirs of infection. Some examples of living reservoirs are animals, humans either being actively ill, so they're suffering from an infection. The microbe can exit their bodies via a portal of exit and enter a new host to be passed on. Or humans can be carriers where they suffer from an asymptomatic, subclinical, or inapparent infection. So all three of these terms mean the same thing. A carrier is a person who has an infection with a microorganism, but they are not aware of it because they don't have any signs or symptoms of the infection. So just like an actively ill human, this living reservoir of the human carrier, the microorganism lives in their body, can exit via a portal of exit and be passed on to another person. Arthropods can also be carriers of infection or reservoirs of infection. So these include all of the insects, and mosquitoes and ticks are big ones. Reservoirs of infection can also be non-living, so we see lots of microorganisms living in the soil, the water, the air, or the built environment, meaning any structure that was built by humans. So one example would be the air ducts in a building. They can harbor microorganisms that would then blow when the air conditioner comes on, and these then can find hosts in humans. There's an additional word in red here, so that means it's a vocabulary word. That word is zoonoses. So any microbe that can infect animals and then pass on to humans is called a zoonoses. So diseases that can jump from animals to humans are zoonoses. The ways that microorganisms travel from one host to another or from one non-living reservoir to a living reservoir can be either through direct transmission. So this is examples of how infectious diseases are transmitted directly from host to host. So we have examples of vertical transmission, which would be just from parent to offspring. So when a mother has a baby, if the mother has HIV, the baby can have a chance of contracting HIV as well, just from being born from this mother. Horizontal infection is when the disease or microbe passes from one carrier to another. They're not directly related, they're just in contact in some way. So some examples of direct or contact transmission are kissing, sex, droplet contact. So if a person sneezes and has the flu is expelled in the sneeze droplets, which another person then inhales. We also have parenteral transmission, which would be transmitting a microorganism from one person to another via injury. So sharing needles for intravenous drug users is a common way that infectious disease is transmitted. If there's a knife that is contaminated with microorganisms and injures a person, that person can then be infected with the microorganism. So these are all examples of direct transmission. We also have indirect transmission of microbes from one host to another. So these are when the hosts are not in direct contact, but their microorganism is shared through some indirect means. So we have fomites, which are inanimate objects that can harbor or transmit pathogens. We have vehicles, which can be air, water, soil, or food. And then we have a special category of oral fecal. So this is what we see in contaminated water sources. If sewage and drinking water become mixed together, then organisms that exit the host in feces can then be ingested when the water is drank, and this can be a way of indirectly transmitting infectious disease from one host to another. Vector transmission involves a arthropod or animal that mediates the carrying of the microbe from one human host to another. A mechanical vector is when the microbe adheres to the outer parts of the host's body, like the legs of a housefly. A biological vector is when the microorganism lives part of its life cycle within the body of the vector. So examples here are malaria, which gets into the mosquito, develops in the mosquito, and then when the mosquito bites a second host, that host gets malaria. Or rabies, which we see getting into animals like raccoons and then developing inside the raccoon's body. When the raccoon bites a person, that person then gets infected with the rabies microbe. Okay, another aspect of disease is the idea that some infections are actually caused by contact with healthcare facilities, which seems counterintuitive. Normally, people go to healthcare facilities to get well, but in some cases, infections can be contracted in the contact with a healthcare facility. So healthcare associated infections, or HAIs, are acquired during a hospital or healthcare facility stay. They're also referred to as nosocomial infections. So on this Venn diagram, we have 3 factors that contribute to HAIs, or nosocomial infections. That is, first, that there's a high presence of microorganisms in the hospital environment, so a lot of sick people and infectious disease, so there's a lot of microorganisms there. A lot of the patients are immunocompromised, meaning their immune systems are not functioning fully because they are sick with some other condition. And lastly, there is transmission of pathogens between staff and patients and among patients. So there's a lot of contact of healthcare workers moving from room to room and patients being transferred. So this can mean that pathogens from one patient can travel to another patient. Standard precautions for preventing HAIs are washing hands, using gloves, personal protective equipment like goggles, face masks, and aprons, safe handling of sharps, so not recapping needles, safe handling of hospital wastes and linens, and then using proper environmental cleanup procedures to reduce the amount of pathogens present in the hospital. Okay, another aspect of disease is what causes it. So the scientist really interested in this question was Robert Koch, and understanding what causes disease is called etiology. So Robert Koch just wanted to figure out what causes infectious disease, and this was long before the discovery of microorganisms. So Koch established a series of steps you can do in order to associate a particular microbe with a particular disease. So first we have a sick individual, and Koch would do his studies on animals because you can't study disease in humans. You can't knowingly infect humans with a microorganism. So Koch would start with a sick animal. He would isolate a sample from the sick animal of blood or tissue. He would then try to culture the tissue to see if any microorganisms were present and if they were growing. And when he grew them in the lab, he would make sure that only one type of microbe was growing. So there's a way of spreading the blood or tissue out in more and more dilutions until you get isolated areas which are all the same type of microbe. So he would do this and he would figure out what microbes are growing in the blood or tissue. Then he would take that microorganism from the lab where he grew it. He would then inject it into a healthy animal, and then he would observe to see if that healthy animal becomes sickened with similar symptoms. Then isolate a sample from the newly sickened animal and try to get the same microorganism to grow. So if he did all of these steps, he would then say that this disease was caused by this microorganism. Lastly, we're going to discuss epidemiology in Chapter 13. So this is the study of disease and how it spreads. So I'm going to introduce a made-up population of 11 people, which are color-coded, with those that were infected, got sick in March, those that were infected and got sick before March, and those that are uninfected. So with epidemiology, we're going to do 2 main concepts. The first is incidence. So incidence is the number of new cases in a population over the total number of persons in the population. So in this scenario, we have 3 people which were newly infected in March. There's 11 total in the population, so we would report the disease incidence as 3 over 11, which you can also divide out into a percentage. So this number is important for tracking new diseases and seeing what's kind of popping up in the population. Prevalence is the number that encompasses all cases of a disease in the population. So in this case, we have the new cases in red, the cases that are older in blue, for a total of 7 people with this disease out of the 11. So the prevalence in this population of this particular disease is 7 out of 11. As diseases are being tracked with incidence and prevalence, we also can look at patterns globally, which is something the World Health Organization and the Centers for Disease Control is charged with doing. An endemic disease is one that occurs constantly in a population at a low level. So an example of an endemic disease would be the flu. The flu always occurs in the population. As long as it stays below a certain level in the winter months, we consider it endemic. Sporadic is a disease which occurs rarely, but it is occurring in isolated areas. So we have small numbers, we have spikes in the amount of people with the disease, so in the amount of incidence and prevalence, but they're pretty isolated, and it's not great numbers. The numbers are still small, although above endemic level. Epidemic is when we have high numbers, very high above endemic level in an area of the world, but it is still in one isolated geographic location. If an epidemic spreads further to multiple locations, it then becomes a pandemic. So a pandemic is when we have global increase in a disease, multiple spots, very high numbers above endemic level. That concludes chapter 13.